Hello everyone. Today we're going to be talking about a qPCR array demonstrates enhanced biodegradation at petroleum impacted sites. Our presenter for today is going to be Dora Ogles with Microbial Insights. For those of you who may not be aware of EOS remediation, we're a small woman-owned firm located in Raleigh, North Carolina. We're the original developers of the emulsified oil technology. We hold patents both domestically and internationally, and we've been globally recognized as a leader in our products and our reputation. I'd like to introduce you to one of our newest products in our lineup, EOS 100. It's a water mixable vegetable oil that's perfect for biobarrier applications, high velocity aquifers, or aquifers that have characteristically low oil retention such as fractured bedrock or highly sandy or gravelly aquifers. It also won the PE Magazine Editor's Choice Award for Product of the Year in July 2013. If you are traveling the conference circuit, please feel free to stop at one of these conferences and visit the booth, RE3 in January, Redox Tech, which is the Southeastern Groundwater Conference in March, and then AHS in San Diego. Without further ado, I'd like to turn it over to our speaker today. Dora, please take it away.
I'm delighted to be doing this webinar. For the presentation, I'm going to be talking about a new technology that's available called QuantArray. I want to show a couple of examples of how this can be used. In the beginning, I'm going to go through some background information and even show several biodegradation pathways. I want to assure you that I'm going to get to data from a couple of sites, so if you can bear with me for a little bit, I want to be sure that everyone's on the same page in the beginning, and then we'll get to this new technology and the specific application of that technology. So the first site is a petroleum distribution facility that's impacted by gasoline. It's currently undergoing monitored natural attenuation, but in situ microcosms were performed to determine whether oxygen addition would stimulate aerobic biodegradation and whether the addition of sulfate would also stimulate anaerobic biodegradation by sulfate-reducing bacteria in two different areas of the site. So when we think about assessing a site for bioremediation, commonly the first line of evidence is to examine contaminant concentrations and look for decreasing trends. However, the decreasing trend doesn't necessarily mean that the contaminant is being biodegraded. As many of you know, decreasing trends can be due to other factors such as dilution and dispersion. In some cases, like MTBE, we can look for daughter product formation. But for a lot of contaminants, intermediates aren't going to be detected in a typical volatile organic compound run. So although this is valuable information, it can still be often difficult to elucidate the mechanisms for some of the compounds. So the next thing that we do is to look at the geochemical evidence. By comparing the parameters like dissolved oxygen, nitrate, ferrous iron, sulfate, and methane in an upgradient versus an impacted well, this helps us to investigate electron acceptor consumption. Geochemical lines of evidence are really good indicators of biological activity, but they're indirect. So one of the direct ways that we can evaluate biodegradation is to look at the organisms or genes that are involved. In the past, we've used plate counts, and we've physically grown these organisms in the laboratory to try and assess the degraders that are present and to what quantity. The problem with this method is that less than 1% of organisms can be grown on plates, either because we don't know how to grow them in the laboratory or because they simply won't grow on that plate. And this can lead us to vastly underestimating bacterial populations. So what we can do is use environmental molecular diagnostic tools such as quantitative polymerase chain reaction, or qPCR, to help us understand and quantify these populations. qPCR gives us the ability to accurately and precisely quantify the organisms without the need to grow them in the laboratory. We can do this by extracting the DNA or the RNA of the organisms and directly assessing that microbial community. The method is very specific and sensitive. Since there can be thousands of different microorganisms present in a sample, qPCR gives us the ability to detect the organism or gene of interest, even if it's less than 1% of the population. It's also very rapid since we don't have to grow it on plates in the laboratory, so we get results in just a few days. So the way that qPCR works is a sample will be collected from a monitoring location and shipped to the laboratory where DNA or RNA would be extracted upon arrival. qPCR can then be used to quantify the organisms or genes of interest. Quickly, the way that this works is qPCR uses short segments of DNA called primers, and those primers will bind to complementary regions which are unique to that organism or gene of interest. It then makes copies of the segment. As it copies across, it displaces a probe that is in the middle of that unique segment, causing a fluorescent molecule to be released. This fluorescence is then measured by the instrument and tracked as more and more copies are made. At the point where the amplification becomes exponential, this is called the cycle threshold for that sample. This threshold can then be correlated back to controls with known concentrations to help us determine the amount of the gene or organism that's in that target sample. The correlation is then reported and then can be combined with those other parameters, that other geochemistry from the site that we were just talking about. So let's say you want to assess feasibility of monitored natural attenuation at a gasoline site like we have here. What we would have done was the sample would be extracted, then individual assays would be performed to quantify the genes of interest. So in this case, we may have picked a couple of assays that we could use to be indicators of aerobic biodegradation, like toluene dioxygenase, the TOD shown, or phenol hydroxylase, PHE. We might also choose to look at an anaerobic indicator of toluene degradation, benzyl succinate synthase, or the BSS. So while qPCR is a great tool and can accurately quantify specific functional genes encoding the enzymes responsible for biodegradation, the question then becomes, which contaminants do we focus on? Petroleum products are mixtures of different classes of compounds. The monoaromatics, benzene, toluene, ethylbenzene, and xylene get the most attention. But there are also fuel oxygenates like MTBE, as well as other PAHs that may be present, particularly for a diesel fuel release. Finally, alkanes are a component of total petroleum hydrocarbons, and these shouldn't be ignored, because actually the alkane content in gasoline can be as high as 20% by volume. So looking at all the possible mechanisms and compounds with individual qPCR assays can provide us a lot of information, but if you try to look at all of them, it can be daunting and a cost-prohibitive task. Not only are there a lot of compounds that can be involved, but each compound can have a variety of biodegradation pathways. Aerobic biodegradation of BTEX has been extensively studied over the last 50 years. Microbes have been isolated, pathways and corresponding genes have been discovered. So if we look at just one component of just BTEX, as like toluene, which is shown here, one degradation pathway begins by the addition of oxygen to the ring, and then it can continue with the second addition of an oxygen. Another aerobic pathway may begin by the oxidation of the methyl side group. And some microbes initiate BTEX biodegradation by simultaneously adding 2 oxygens. The common denominator is that all of these aerobic pathways are initiated by oxygenase enzymes, and we can use qPCR to target the initial oxidation enzymes of these pathways to quantify the bacteria that are capable of BTEX biodegradation. But we also have to consider that BTEX compounds may be susceptible to biodegradation under anaerobic conditions. And all the biodegradation pathways for these compounds haven't been as well characterized as the aerobic pathways. We still know several of them that we can target. So as shown here for toluene, the first step in the anaerobic pathway is the addition of fumarate onto the methyl group to form benzyl succinate. This initial activation is mediated by a gene called benzyl succinate synthase, and that's the one that I just showed in the last slide, BSS. So in total, there are 6 known pathways for just biodegradation of toluene, 5 aerobic, 1 anaerobic. All, several, or none may be contributing to toluene biodegradation. So how do we choose the appropriate targets if we can only do a few qPCR assays? Well, the short answer is now that we have this new technology, we won't have to do that. QuantArray is a platform that can be used to target numerous pathways all in one analysis. It gives us the same accurate quantification as qPCR. The way that it works is very similar to what you just saw for the qPCR, that the sample's collected, DNA or RNA is directly extracted from the sample. Then rather than running individual qPCR assays for each target, the subarray platform will give us the ability to run large amounts of assays. So what you see here is an example of the subarray. This would be one sample that would be added here, and each circle that you see there is a through hole that contains the necessary components for a qPCR reaction. The through holes have hydrophobic and hydrophilic components that allow that reaction to happen in that little through hole. So it will allow us to generate a curve from each of those little holes showing the concentration of the gene or organism that we're targeting with primers and probes that are specific for that in each through hole. So the results from this, rather than giving us individual curves, will give us up to 56 different qPCR assays, all from that one analysis. So as promised, I'm getting a little more closely to what we're going to be showing you data from the site. This is the list of the different genes that we quantified for this site using the QuantArray Petro. The gene targets in the table are grouped by contaminant and redox conditions. The top section includes assays for quantification of monooxygenase and dioxygenase genes responsible for aerobic biodegradation of monoaromatics. We also look at methylibium petrophilium PM1, which is an organism that's important for MTBE biodegradation, and then at a TBA monooxygenase. Another section is specific to the anaerobic degradation of the monoaromatics. And I'd like to make mention of a gene that we think is going to be important. It's recently identified, and it's an anaerobic benzene carboxylase gene. To date, this is the only known pathway for the anaerobic biodegradation of benzene, and you'll see some examples of how this was important at this site. The Petroleum QuantArray also encompasses genes for aerobic and anaerobic biodegradation of PAHs, as well as aerobic and anaerobic biodegradation of alkanes. And finally, we can look at the response of total bacteria and sulfate-reducing bacteria. These groups are very helpful, especially when we're going to be adding some type of electron acceptor, so we can see how the electron acceptor is impacting a specific group that we're interested in or that total bacterial population. Okay, so finally, as promised, let's get to that data from the facility. Again, it's a petroleum distribution facility where the groundwater is impacted by gasoline. We used in situ microcosm studies to look at the site with and without the addition of sulfate as an electron donor. So for those of you that are not familiar with in situ microcosms, these are slotted PVC that have baffles on each end. The baffles allow us to segment off different sections of the monitoring well so that we can answer questions in situ. Inside each unit is the following. First, we have a passive diffusion bag that we can use to collect the volatile organic compounds. Second is a biotrap sampler. And a biotrap contains a special matrix called BioStim that's composed of powder-activated carbon and Nomax. So the biotrap will act as a passive sampler for the microorganisms that are present, giving them a place to attach and grow. Finally, in each unit, we have a small vial that we can use to collect the geochemistry. And what we did for this study is we had 2 units that were deployed in each monitoring well. The first unit was a monitored natural attenuation unit that we could use as a control to understand natural conditions at the site. The second unit was a biostimulation unit where we wanted to add the electron acceptor to see what's going to happen. So we used EAS, which is a product offered by EOS remediation. It's a sulfate solution that takes advantage of the pre-existing anaerobic conditions to help us promote biodegradation. So when we look at the site, there were 2 wells that were selected in the source area. These wells had anoxic conditions. We have MW23 and MW19S. So in situ microcosms were installed, and there was an M&A unit and an EAS amended unit that was in each monitoring well. So you're going to see 4 different units that we're going to look at the data from. So what we have here are the results obtained from the M&A unit, so looking at the natural conditions at the site. The y-axis shows us the concentrations of the gene, and then along the x-axis, you'll see genes involved in the anaerobic biodegradation of hydrocarbons, and we'll talk about each of these a little bit. But I want to start by looking at the last gene that's shown here, APS. APS is adenosine phosphate synthase, and this is a gene that's found in sulfate-reducing bacteria. We see this represented by the 2 blue bars, so we see that we had detection in both MW19S and MW23. So sulfate reducers were there under the existing site conditions. But of the anaerobic functional genes that were responsible for BTEX and PAH degradation, we don't see any of these detected. They're below detection limits in the control unit. In the biostimulation unit, which you see with the red bars that come up here, sulfate-reducing populations are approximately an order of magnitude greater than what we saw in an M&A unit. Moreover, BCR genes, which had not been detected in the M&A unit, were 10 to the 3 cells per bead in the EAS amended unit deployed in MW23, and 10 to the 5 cells per bead in the EAS amended unit in MW19S. Benzyl coenzyme A is a central intermediate in the biodegradation of many aromatic hydrocarbons, and this benzyl coenzyme A reductase gene that we have here shown as BCR is an essential enzyme for reducing the benzene structure. So when we look at other anaerobic genes, we see that benzyl succinase synthase genes encoding the enzyme responsible for initiating the anaerobic biodegradation of toluene and other alkyl substituted benzenes were detected on the order of 10 to the 3 cells per bead in the EAS emitted unit in MW19. We did not see this come up in MW23. We look at ABC, and if you remember, this is a gene that I mentioned earlier. This is the anaerobic benzene carboxylase gene. It was detected in the EAS amended unit deployed in monitoring well MW23, suggesting that EAS addition promoted growth of bacterial populations capable of the anaerobic biodegradation of benzene in this well. So there are two other genes that you see listed here, NMS and ANC, and they weren't detected in either the natural units or in the EAS amended units in either well. NMS is naphthyl methyl succinate synthase and is analogous to the BSSA gene that we just described for toluene, but it catalyzes the addition of fumarate onto the methyl group of 2-methylnaphthalene. Then we also didn't see ANC, which is an anaerobic naphthalene carboxylase gene, and to date it's the only pathway that's been characterized for the anaerobic biodegradation of naphthalene. So when we look at the data, the first question that we ask is, why was ABC detected in the BioStim unit deployed in 23 but not in 19, and vice versa? Why are we seeing BSS in 19 but not 23? Well, actually it corresponds really well to what we saw with the contaminant concentrations. At MW23, where the ABC genes were detected, benzene was the predominant BTEX compound present, while at MW19S, the alkyl substituted benzenes, ethylbenzene and xylene, which serve as substrates for the BSS enzyme, were the predominant volatile organic compounds. So what did the study show? What was the impact of biostimulation with EAS? As might be expected, we saw an increase in sulfate-reducing bacteria. More importantly, though, there were significant increases in concentrations of genes involved in the anaerobic biodegradation of BTEX. So BCR, which is a common intermediate for anaerobic metabolism of aromatics. We also saw an increase in MW19 of BSS, which initiates anaerobic biodegradation of the alkyl substituted benzenes by the fumarate addition. And finally, in MW23, where benzene was the primary contaminant, EAS addition stimulated growth of bacteria harboring those anaerobic benzene carboxylase genes. So we have anaerobic benzene metabolism. So EAS addition stimulated growth of anaerobic BTEX degrading bacteria. That's really what we wanted to see, so that's great because BTEX was the primary contaminant of concern. But when we looked at the data a little closer, we didn't see any indication that sulfate addition stimulated the growth of anaerobic PAH and alkane degraders. There could be a couple of reasons for this. First, the deployment period was about 50 days, which isn't unreasonable for this type of study, but it's certainly shorter than the lifespan of a full-scale injection. The other factor to consider are the concentrations. PAH concentrations in these locations range from 7 to 160 parts per billion, whereas BTEX concentrations are much higher, 2,300 to 3,200 parts per billion. So an additional in situ microcosm study was also performed at MW7, and this well is located further up gradient of the main source area. Contaminant concentrations here are lower, as would be expected, and conditions are already mildly aerobic. The question was DO really a limiting factor? And if we add oxygen, would that stimulate biodegradation of the remaining contaminants? So we had another in situ microcosm that was set up for this well, and it was the same as before, where we have a control unit to look at the natural conditions and a biostimulation unit. And what we see in the biostimulation unit was rather than using the EAS, we used an EOX, which is a powder that provides slow, steady oxygen release to help us get long-term aerobic biodegradation. So again, let's look at the control unit first. Concentrations are again shown on the y-axis, and the x-axis we have a lot of genes listed here. We have aerobic genes for BTEX degradation. These are RMO, RDEG, PHE, TOD, and TOL. These are all genes that are expressed during biodegradation of BTEX. Then we looked at EDO, which is an important gene for ethylbenzene and isopropylbenzene biodegradation. We also looked at BPH4. In environmental restoration, biphenyl dioxygenases are best known for the cometabolism of polychlorinated biphenyls, or PCBs. However, the subfamily also includes benzene and isopropylbenzene dioxygenases. PM1 is the next target. If you remember, we discussed this earlier. This is Methyllibium petrophilium, and it's one of the few bacteria that's capable of growing on MTBE. So when we see this gene, it's indicative of the potential for aerobic MTBE biodegradation. We also looked at TBA monooxygenase and naphthalene dioxygenase. And as you can see, we have quite a few of these genes that were detected under natural conditions. Since conditions in the well are already mildly aerobic, it really isn't surprising that we're seeing these aerobic genes present. So what happens when we look at the addition of oxygen? We do see that this increased concentrations of several genes that are important for aerobic biodegradation of BTEX and naphthalene. And one gene that I'd like to really mention is this toluene 2 monooxygenase gene, RDEG here. This is a gene that wasn't commonly selected for standard qPCR analysis. So if we were solely doing qPCR at this site and we only had limited budget to do a few genes, we may have missed this pathway that was actually stimulated by the oxygen addition. So to summarize this study, what we saw is under natural conditions, there were substantial populations of organisms that did have the genetic potential to degrade BTEX, MTBE, and PAHs. When we add oxygen, we see increases in the genes that are important for BTEX biodegradation as well as naphthalene biodegradation. So in general, the QuantArray technology was used to evaluate conditions across the site, and we were sure that we didn't miss pathways that could be important since we were looking at so many, all the known ones. As we saw in the aerobic study, a toluene monooxygenase not commonly analyzed by standard qPCR was one of the genes stimulated. Then when we looked at the electron acceptor addition, the QuantArray Petro was used to show the impact on the response of the microbial populations. So from this study, the assessment of MW19S and 23 close to the source zone showed that the addition of EAS stimulated anaerobic growth of BTEX-utilizing organisms. The study at MW7 showed that baseline degrader populations were already significant, but we still saw increases with the addition of oxygen. We could further evaluate this study and perform it using the active expression of these genes using RNA from the cells rather than the genetic potential as we did here for DNA. Microorganisms with the genetic potential to degrade BTEX are fairly ubiquitous in the environment. However, they may not be performing the function that we're interested in because most of them have pretty variable metabolic capabilities. So we could perform additional studies using RNA to see if the organisms present were actually expressing those genes. And then we could look at the oxygen amendment to see how much the expression of those genes was stimulated by the available oxygen. So now I want to switch tracks just a little bit, and since this is a new technology, I also wanted to show another quick case study of where the QuantArray could be used, but for chlorinated solvents. This is a site with PCE and TCE are the primary contaminants, but trichlorobenzene and dichlorobenzenes are also present. So the client wanted to consider their treatment options and perform an in situ microcosm study in lieu of a pilot study. As many of you know, anaerobic biodegradation of chlorinated solvents occurs via the pathways shown above. We have sequential reductive dechlorination of PCE and TCE through DCE to vinyl chloride and ultimately ethene. Several groups of organisms are capable of reductively dechlorinating PCE and TCE, as we have shown here. We have Dihalococoides, Dihalobacter, Disulfidobacterium, and Disulphiromonas. But there's only one known organism that can dechlorinate the DCE and vinyl chloride, Dihalococoides. And we can also look at a couple of functional genes to make sure the vinyl chloride to ethene conversion is going to be an efficient step. For chlorinated benzenes, the rule of thumb is that trichlorobenzenes, dichlorobenzenes, and chlorobenzene are more amenable to aerobic biodegradation and can serve as growth-supporting substrates. However, there has been evidence indicating that complete reduction of trichlorobenzenes is possible. Dihalococoides strain CBDB1 has been shown to dechlorinate 1,2,4-trichlorobenzene to a mixture of dichlorobenzenes, but they were not further degraded. More recently, however, Nelson et al. Reported sediment microcosm results where dihalobacter were implicated in the reductive dechlorination of dichlorobenzene to chlorobenzene and ultimately to benzene. Under aerobic conditions, several different types of bacteria, including methane oxidizing bacteria or methanotrophs, ammonia oxidizing bacteria, and some toluene phenol oxidizing bacteria can utilize or co-oxidize trichloroethene, dichloroethene, and vinyl chloride. So in general, cometabolism of chlorinated ethenes is mediated by monooxygenase genes with relaxed specificity that oxidize a primary growth-supporting substrate and co-oxidize the chlorinated compound. So like in this example, in the presence of methane, methanotrophs produce methane monooxygenases, which will oxidize methane to methanol, but can also co-oxidize the TCE. So just like the methane monooxygenase, aerobic oxygenases have also exhibited relaxed specificity, and many are capable of co-oxidation of TCE and lesser chlorinated ethenes. The example shown here is for toluene dioxygenase, which has received the most attention for aerobic cometabolism of chlorinated ethenes. It certainly isn't the only one that can do it, however. Each of the toluene monooxygenases, or more specifically the ring hydroxylating monooxygenases like toluene 2, 3, and 4, have been shown to be capable of co-oxidation of TCE. So when expressed, toluene dioxygenases and monooxygenases may be contributing to the cometabolism of TCE, particularly at mixed waste sites where TCE and BTEX, or in this case chlorinated benzenes, are co-contaminants. Okay, this is the final pathway slide, I promise. As I mentioned previously, trichlorobenzenes can serve as growth-supporting substrates. The aerobic metabolism of 1,2,4-trichlorobenzene has been elucidated in a number of pseudomonads and is initiated by a trichlorobenzene dioxygenase gene. This is a unique group of trichlorobenzene and even tetrachlorobenzene dioxygenases that are distinct from the toluene and benzene type of dioxygenases that we described earlier. So this time for the in situ microcosm study, we had 3 units that were installed in the monitoring well. One was to look at the natural conditions of the site, one to look at an electron donor addition, and finally one to look at the impact of a bioaugmentation culture. Inside each of the units was a biotrap sampler to look at the microbial community, a passive diffusion bag to look at the contaminants of concern, and as we showed before, a vial to look at the geochemistry. The biostimulation and bioaugmentation units had sponges on each end containing electron donor. And it's important to note that the bioaugmentation unit, we had a biotrap that was loaded with a known concentration of a commercially available dihalococoides culture, was loaded onto the bioset beads, and this was deployed in the unit. Due to the 2 groups of contaminants present at the site, chlorinated ethenes and benzenes, the number of potential degradation pathways, QuantArray was going to be simply the most cost-effective method to help us understand the microbial communities. So as mentioned before, a number of different genera of bacteria may be contributing to reductive dechlorination of PCE and TCE. So with the QuantArray, we can quantify these for more comprehensive assessment. Dihalococoides, along with functional genes for reductive dechlorination of vinyl chloride, are also included to help us evaluate whether complete reductive dechlorination of ethene is likely to occur or whether biodegradation may stall at cis-DCE. For this site, we are also interested in the potential for the anaerobic biodegradation of chlorinated benzenes. dihalococoides strains like CBDB1, which are capable of reductive dechlorination of a variety of chlorinated benzenes, including the trichlorobenzenes, as well as the higher chlorinated benzenes like tetra, penta, and hexachlorobenzene. So we wanted to look at that organism, as well as the halobacter, which has been linked to the chlorination of dichlorobenzenes and chlorobenzene. So running through the anaerobic cometabolic pathways quickly, the QuantArray includes quantification for both types of methane monooxygenase genes, also toluene dioxygenase and the ring hydroxylating monooxygenases that we described earlier. And while I didn't mention this pathway earlier, this is also on the QuantArray chlor, that depending on subsurface conditions, aerobic ethene-utilizing bacteria may also contribute to vinyl chloride degradation. The initial steps in this pathway are catalyzed by an ethene monooxygenase and a transferase with mediate formation and a breaking of the epoxide. So consider a site where active reductive dechlorination is occurring in the source zone and producing vinyl chloride and ethene. In more aerobic, down gradient portion of the plume, ethene and vinyl chloride oxidation may also be an important mechanism that we should consider. Finally, we can look at total bacteria as well as other functional groups. For example, both sulfate-reducing bacteria and methanogens can potentially compete with halo-respiring bacteria like dihalococoides for available electron donor. So following electron donor addition, in fact, we have seen commonly that there's a temporary increase in sulfate-reducing bacteria prior to seeing an increase in dihalococoides. So returning to the chlorinated solvent site, this is a slide showing the data from the monitored natural attenuation unit, so our control unit, and I've plotted it in a really simplistic fashion. What we try to use here is what my friend Steve Koenigsberg likes to call a red light, green light approach to the numbers. What we see is that within the M&A unit, there are low to mid-range concentrations of organisms important for anaerobic biodegradation of chlorinated ethenes, ethanes, and chlorinated benzenes. There are also low concentrations, with the exception of one gene, involved in the cometabolism of chlorinated ethenes and chlorinated benzenes that were observed. So if we look at this a little more closely, again, we're still looking at the control unit with concentrations of genes or organisms on the y-axis. First, we're looking at dihalococoides and the functional genes that are known for dihalococoides, TCE reductase, as well as 2 vinyl chloride reductase genes. Then Dihalobacter, Dihalogenamonas, Disulfidobacterium, Dihalobium, and Disulfiramonas, which can be involved in reductive dechlorination of some of the higher chlorinated compounds. The last gene you see here, 1,2-DCP, is an important gene for chlorinated propanes. And specifically, we found that under natural conditions, there were low to moderate concentrations of Dihalococoides and Dihalobacter. We also saw that the TCE reductase gene found in Dihalococoides was present. But it's really important to note that for Dihalococoides, neither of the vinyl chloride reductase genes were detected. So we're likely to see an accumulation of vinyl chloride at the site. When we look at the contaminant concentrations from the passive diffusion bag within the M&A unit, we see that the primary detection was DCE and some TCE, and only a little bit of VC and ethene. This correlates well with the microbial data, since we saw low concentrations of Dihalococoides and Dihalobacter, and vinyl chloride reductase genes were below the laboratory detection limit. So shifting in the same control unit to look at the aerobic biodegradation potential, we did not see the trichlorobenzene dioxygenase gene, suggesting limited potential for growth-supporting aerobic biodegradation of the trichlorobenzene under existing conditions. When we looked at genes associated with aerobic cometabolism, results were mixed and not overwhelmingly encouraging. Although toluene 2 monooxygenase and phenol hydroxylase genes were present, RMO, toluene dioxygenase, methane monooxygenase, and even the ethene catabolic genes were all below detection limit. So in summary, we see that under natural conditions, there are low to mid-range concentrations of Dihalococoides and Dihalobacter, but vinyl chloride reductase genes were not detected, indicating that cis-DCE will likely continue to accumulate. The trichlorobenzene dioxygenase gene was not detected, suggesting limited potential for the aerobic biodegradation of trichlorobenzene. And although we saw a couple of aromatic genes which are capable of the cometabolism of TCE, other common cometabolic genes were below detection. So overall, the results from the M&A unit suggest that this may not be feasible, or at least not practical, given the time constraints that there were at the site. So what happened when we added an electron donor? What you see here, the blue bars show our monitored natural attenuation or our control unit, and the red bars are for our biostimulation unit. We see that there's a higher concentration of Dehalobacter in the biostimulation unit, 3 orders of magnitude higher. And although we see that Dehalococcoides concentrations are similar, we're now detecting low concentrations of the BAV1 vinyl chloride reductase gene found in some strains of Dehalococcoides. This gene is really important for the degradation of vinyl chloride. So what do we see looking at the contaminant concentrations? We see that the biostimulation unit is almost all cis-DCE. So it certainly seems that electron donor addition enhanced reductive dechlorination of TCE. While certainly not conclusive, there also was an increase in vinyl chloride concentrations, even though it was a small one. We also looked at contaminant concentrations for the chlorinated benzenes in these 2 units. The predominant contaminant in both the M&A and BioStim units was 1,2,4-trichlorobenzene. The biostimulation unit had a lower concentration of the 1,2,4-TCB, while dichlorobenzene concentrations were higher than in the M&A unit, suggesting the potential for enhanced degradation. However, the difference in TCB concentrations was only 4%, and the daughter product formation was inconsistent with literature reports. Daughter products from 1,2,4-trichlorobenzene dechlorination are predominantly 1,4-dichlorobenzene about 70% of the time, and 1,3-dichlorobenzene about 30% of the time. So preferential removal of a flanked chlorine. In this case, the primary difference was in concentrations between the M&A unit was in the 1,2-dichlorobenzene rather than the typical products for the 1,2,4-trichlorobenzene dechlorination. So in terms of enhanced trichlorobenzene biodegradation, it's happening perhaps, but it's certainly less conclusive than what we saw for TCE. So looking at the overall results from the biostimulation unit, we see that Dehalobacter populations increased substantially with the addition of electron donor, and one of the vinyl chloride genes was now also being detected. Combined with the enhanced production of cis-DCE, we see that biostimulation may be feasible for the reductive dechlorination of TCE. So what happens when we add a bioaugmentation culture? So what you're seeing here, the red bars are from the biostimulation unit, the green bars are the bioaugmentation unit, and we see that concentrations of Dehalococcoides and vinyl chloride reductase genes are much higher, indicating that the culture was able to survive in situ during that deployment period. Dehalobacter and Halogenemonas were also detected in this bioaugmentation unit. So when we look at the contaminant concentrations of the chlorinated ethenes in the bioaugmentation unit, we see that cis-DCE concentrations are lower, and there's increased production of vinyl chloride and ethene, which is consistent with the higher Dehalococcoides populations and the greater concentrations of vinyl chloride reductase genes. But when we look at the chlorinated benzenes, despite the high concentrations of Dehalococcoides, concentrations of these were not impacted by the addition of the bioaugmentation culture. So in conclusion, observations from the bioaugmentation unit show that Dehalococcoides and vinyl chloride reductase genes were 3 orders of magnitude greater than with biostimulation alone, and there was enhanced vinyl chloride and ethene production during the deployment period. So bioaugmentation may appreciably enhance reductive dechlorination of TCE. At least within the deployment period, however, bioaugmentation did not have an impact on the chlorinated benzenes. So from these 2 case studies, we're able to see that the QuantArray technology was able to help us understand the potential for monitored natural attenuation, being sure not to miss an important pathway, and we can use this information to help us understand which pathways are most dominant at a site so we can select the most appropriate electron donor or acceptor to add. Then once those donors and acceptors are added, we can use the QuantArray data to help us understand the impact that they're having on the microbial community at the site. So at this point, I'll be glad to take any questions that you have.
Excellent. Dora, thank you so much for that. I just want to apologize again to the audience for the technical difficulties we had at the beginning. We are recording this presentation and we will make it available on our website. So let's jump into some questions. Dora, I think some people maybe missed the first couple of slides of your presentation, so I got a couple of questions here. Could you just briefly go over exactly what qPCR is, kind of what the acronym stands for, and sort of the methodology behind that?
Sure. So qPCR is quantitative polymerase chain reaction. And the way that it works is you have short segments of DNA called primers and another short segment of DNA called a probe that has a fluorescent marker on it. And so the primers and probe will bind to a unique region that we're interested in for that organism or that functional gene that will help us be sure we're only targeting that organism or gene. And the process for polymerase chain reaction is where it'll make copies of that gene, and as it becomes exponential, like 2 copies become 4, become 8, then that becomes the cycle threshold of that sample. Then we can compare that cycle threshold value back to known values from concentrations of control organisms that we've analyzed to know what the concentration is in that target sample.
And if folks out there are interested in kind of what the typical cost is between the QuantArray or just doing the general qPCR analysis, they should probably contact you directly?
They can, or I can give you kind of a ballpark view. Quantitative PCR costs $275 for the first target that we analyze on a sample, and then $75 for each additional target. So you can really accumulate a lot of cost if you're looking at all these different organisms and genes. The QuantArray is $750 a sample, so you get a lot more information for that value.
Gotcha. Earlier in the talk, when you were describing, I think, the BTEX degradation using, I believe it was the EAS, you had mentioned in your summary from that step of the pilot that you saw, the amendment was actually stimulating the bacteria there, and you saw a substantial increase. Could you quantify that? Were you seeing like a 1 order of magnitude increase, 2 orders? Like, what would you define as a substantial increase in the population?
Some of them went from below laboratory detection limits of those genes to 10 to the 3 to 10 to the 5 cells per bead. So an obvious change. There's no mistake that the addition of sulfate caused growth of those organisms within that unit.
Excellent. So after the addition of a bioaugmentation culture, you saw a reduction in D. halobacter concentrations. Could you explain this, or how would you explain that phenomenon?
After the addition of the bioaugmentation culture?
Yeah, I think
it may be that yeah, it may be that bioaugmentation culture did not have D. halobacter in it. So it was just whatever was naturally that colonized that biotrap during that deployment period. But it obviously had some dihalogenamonas in that culture because we did not see that in the biostimulation unit or the control unit. We only saw it when the bioaugmentation culture was added.
All right. That is all the current questions I have on my screen. If anybody else has a question, please feel free to type it in. Going back to the question about the BTEX stimulating the microorganisms, you said there was several-fold increase in cell concentrations. Can you say with confidence that a 1 to 3 order of magnitude increase in gene count is from the addition of the electron, either donor or acceptor, and not from a natural variation?
There could be some natural variations in the monitoring well, so it is possible that some of that could be, but I would not think we would see that much variation because we're only talking about 15 inches in length for these units, so it's not a huge difference in depth.
All right. All right, a couple more questions popping up. is there any bacterial species that you found most effective in biodegradation? And this question is not specific to any type of contamination, but maybe drawing from your experience, Dora, do you see any one particular species out there that seems to be very effective for degrading, any kind of contaminant out there?
Well, it really depends on the contaminant, because most organisms have certain metabolic capabilities, and so they're not able to use kind of a very diverse group of contaminants typically. So most of the aromatic BTEX utilizing bacteria are probably the most diverse in their capability. These are Pseudomonas and Rhodococcus that have a lot of metabolic functions, and that's why we need to look at these functional genes to make sure that they're actively making more copies of that gene that we're interested in. But as far as chlorinated solvents, the Halicocoides is probably the most important organism known to date. Nothing else has, the 12 years that I've been doing this, there's not been another organism that's isolated that can do this phenyl chloride to ethene conversion step that's important.
Right. Okay. If there seems to be some questions popping up that seem to be pretty specific, I think it might be best, Dora, if you give out your contact information. It looks like people have some various projects and things like that. One gentleman wants to understand, if he has existing qPCR data, is that something that he could share with you and help maybe get some recommendations or some interpretations on that data?
Sure, yeah. I mean, feel free to contact me. My email address is dogles, D-O-G-L-E-S, at microbe, microbe.com. You can also contact Brad. He has my contact information. But, yeah, we all the time are asked to help people understand what do these concentrations mean of organisms. Is this high, medium, or low compared to what you've seen with other sites? Does this look like a good potential for whatever type of stimulation that we're going to be trying? And we can definitely help you with that or scope out further studies that may be able to address those questions.
Great. All right. Well, it looks like we're about out of time. Again, Dora, thank you so much for your time in presenting on this topic. It was very interesting. I know I learned a few things today. It's a fantastic presentation. If you have very specific questions and you need to get in direct contact with myself or Dora, my contact information is there on the screen. Please feel free to email me or give me a call. I'll be happy to help you out. Just one more time, just thank you so much, Dora, and everyone out there. I really appreciate your attendance. Have a great day.
All right.